but I guess for you it's like 73 cubic inches roughly. So it contains enormous number of the nerve cells, 100 billions of them. It's a huge number. So this is, most of them are actually neurons, and that's how neurons looks like. It's a cell, has a body, has essentially a long axon, have a dendrites, and has the neuron density. It's in the gray matter, which I'm going to talk very shortly, roughly is like 50,000 of neurons packed in 1 cubic millimeter. So in very small area of the brain, like somewhere here, there's those neurons are packed, and not only neurons but other cells. There's a lot of stuff in there, and I'm going to show you a little bit about this. But fascinating part of this is this: each neurons can make a lot of connections, a lot of them. So like say 10,000 connections with other neurons. So imagine how complex this is. You have one neurons, it can connect with many, many other neurons. And we have billions of them, and they form connections. And those connections are not static, they develop. They are active constantly during our lifespan. So clearly, this is by far the most complex organ in human body. No wonder it also consumes quite a bit of energy, like 20% of body energy. So there's really 3 main tissue in the brain. I'm not going to give you anatomy lesson of the brain. There's a lot of important regions. I mentioned some, but I'm not going to mention the brain anatomy in details. But essentially there's gray matter, which on this image, this is image from MR, by the way. So you can tell this is the human head, right? So eyes would be somewhere here. This is part of the nose. This is back of your brain. This is front of your head, right? So you can see the gray matter is actually, this image is really gray. So it's like this. So this is the gray matter. And gray matter is nerve tissue, mostly composed of the nerve cell bodies and dendrites. So mostly neurons are packed there. And we also have a white matter. So this is like white stuff here, So you don't really see too much structure within the white matter. But white matter is on this image. White matter is also nerve tissue, mostly composed of a long range of nerve fibers. It's a maybe fancy word, but essentially you can think about it like a tube. So the white matter is like a highway connecting the essentially different areas of the gray matter. So essentially this myelinated is like a cover around the axon, which allows to faster transmission of the signal. So, and also there is actually liquid in the brain. It's cerebral spinal fluid. So on this image, it appears as a dark void space, but it's not void, it's filled with liquid. So also you see it here, you see it here, you see it here. So it actually has a very important role. It's essentially like a protections, because we got hit, and there's like a mechanical protection, so it helps us to protect the brain from the damage, and also provide immunological protection. So essentially you can think, because the brain, there's a lot of chemistry going in the brain, and there's also lots of byproducts. So it's essentially wash out the stuff, which is not sometimes good for you. And if you are healthy, it's pretty much, does not have anything in there. There's almost like water, not exactly, but I mean, it's just, so if you have some proteins in the CSF, and CSF is contaminated by larger molecules, that actually may be a sign of the disease. So, but again, white matter on this image appears to be boring, right? You don't really see much. But actually, with MRI, we can map those fibers in the white matter. So I'm going to show you a very colorful image now. And this image is 2-dimensional image. So roughly you can see correspond to the previous image. So the eye would be somewhere here. So this is front of the head. This is back of the head. So this image actually shows the structure within the white matter. It's a 2D image, but the white matter fibers are not aligned only on those planes. They are going outside, they're going top and down, they're going left and right, And this image essentially is like the projections, and the direction on the fibers, in white matters are encoded as the colors. So essentially, on the previous image, the white matter stuff looks boring. This image looks gorgeous and shows you how complex actually even white matter is. So, we have a brain, lots of species, they also do have a brain. Brain evolved for many, many years, as a species we evolved. So, human brain is shown here. There's, other species' brains are shown here. So even mice has a brain, right there. So, it's just, and those brains are obviously, they are not in vivo brains, so they are ex vivo brains. So some, this individual donate the brain to the science, so essentially it was extracted and we know how it looks. And also in animals, we can extract the brain and we can actually then cut the brain into the pieces and then see, at very high resolution how it looks. So I'd like to show you very small piece of the mice brain. And there is a recent advance in the essentially imaging of postmortem brain tissue. And this image shows you 1 millimeter or 0.04 inch of the brain tissue from the mice. And this image shows you how neurons are packed and how many of them are there. I mentioned on the first slide, there's like 50,000 neurons per millimeter cube on the human brain. This is mouse, a little bit less, but still a lot. So all those green, red dots, they are essentially neuron cells, and they are packed. There's so many of them. So again, basic facts about the brain. So because it's, takes a lot of energy, so there's a lot of blood inside the brain, actually. So received 15% of the cardiac output means there's a lot of blood coming to the brain and then leaving the brain. And there's actually, because the brain needs essentially energy, so there's brain consumes a lot of oxygen, like 20% of the oxygen consumption. And for the energy to be produced inside the brain, the essential sugar is needed. So it's essentially glucose utilization is 25% of our total glucose utilization. And brain requires about 10 times more energy to function properly than other tissue in our body. And really, living brain is never addressed. You are listening to me now, I'm talking to you, so my brain, is active. I got exercise, I went to my office to bring the power supply, got active. You're a little bit maybe bored, but your brain is still working. You go to sleep, your brain is working. You cannot turn off the brain. It's always working. So because brain needs energy, sugar is needed, right? So the energy is produced in the presence of the oxygen. So how the oxygen is delivered to the brain? Inside the brain, we have vast network of the vessels. So this slides, is showing you, again, I select very small, we call it voxel, from the brain, from the gray matter, like say 2 millimeter in size, so twice as much as I gave you from the mice. But besides, neuronal cells, like neurons, which are packed there, there's also like a forest of the vessels, essentially. So here's the blue vessels are essentially, the red vessels are arteries, which delivers the brain. The blue are, the blood is coming out from the brain. Here you can see essentially on this, on the MRI voxel size, that you have like a very small capillaries which penetrates the gray matter. So they are actually very close to neurons. So, this is the way how the neurons get actually oxygen, because the small capillaries are nearby. So, essentially the brain is very complex and multi-scale organ. and it operates on many different spatial and temporal scales. So this actually shows you, for example, if you look on this, so this is our body, right? So we know the size of our body. Our brain is right there. So the spatial scale of our body is, our height, our surrounding, right? So now we go a little bit, we reduce our view. So let's focus on the head and on the brain. So we're talking about the centimeter inches, right? Then we can go actually, even deeper. So we can look on the certain part of the brains, certain regions of the brain. So our spatial scales is even less and less and less. And then we can, go even more. So we can look on the, actually, we call it circuits. It's essentially different brain regions. One, for example, maybe here, another maybe here, very inside the brain. They interact with each other, form the circuit. So this is even smaller scale. But also there is a temporal scale, because essentially we live for the certain age, right? So we're talking about the years, right? So then essentially, our behavior, we're talking days, hours, and then we have essentially, we can look on also essentially minutes and seconds, depending what process in the brains we would like to look at. And then we can go even further down in space and in time. So as you can, what I'd like to show you, that essentially complexity on the brain is not only on the spatial scale, but also on the temporal scale. So we know that brain is structurally and functionally organized. And we call them, we call this cortical areas because, say, this is front of the brain. So this part of the brain, it's, we know now by many years of research of the different fields, neuroscience and, anatomists and monkey and animal studies, that essentially, and in humans particularly, this part of the brain is responsible for cognition and social function. The back of the brain, right there, is essentially responsible for the visual information. So it's called visual cortex. You're watching the slides, and if I would put you in the MRI machine, for example, you and you, this part of the brain will be activated. And I will show you how this activation looks. So this is actually, how the brain processes visual information from the eye. It goes through the eye, goes actually from another parts of the brain, and then ends up here. And brain is actually processing this information from the vision. This part of the brain is language, reading, speech. So when you go to school, learn how to read, and also you speak, this part of the brain is actually activated. There's also motor area in the brain. So I'm moving, you move your head, you move your finger, you move your legs, you're walking, so this part of the brain is activated. And so on, so on, so on. So we know that brain is really organized in cortical areas. But also we know that actually another level of organization is actually in networks of areas. So what this simply means is that, when brain processes certain information, not only one region or one cortical area is activated, but they may be co-activation elsewhere. So they form networks. And that's what we believe now, how brain is actually operating. So there is not brain does not necessarily have a special area for everything. I found something, there must be special area in the brain. Yes, the answer is yes and no, but in fact, the brain, how it works, how we believe it works, it essentially operates as a network. So the different brain regions exchange information with each other, and this information flow, for example, is affected and abnormal when the people develop psychiatric disorders. Okay, so, that was the introduction to our journey goal, which is the brain. So now how we can actually non-invasively make those pictures and images. And that's next few minutes I'd like to show you how magnetic resonance imaging techniques really works. Well, we need some equipment to do so, and it's called MRI scanner. So how it looks, basically, looks like this, almost. So this is almost whole MRI scanner. I'm going to show you later on other pieces of it. But essentially, we have like this thing is like a big magnet. So this magnet is constantly on. So magnetic field is generated actually inside this magnet. So then we can put the patient in, as you can see. And then essentially this thing can generate some noise. Like, for example, I'm not sure if you're going to hear it, but let me see. Maybe. Let's see. Well, if someone would call me, you will hear how the scanner sounds. But anyway, there is actually noise generated by the scanner, so that's why when we put the subject in, they actually hear we can put some earplugs, and the noise is not, it's not dangerous, but some people don't like it. For some people it's annoying, so we can actually give you headsets and put the earplugs so you can listen to a movie, watch the movie. So it's actually, some people don't even notice that there's a noise, but actually for some experiments the noise is noticeable. So bottom line is, this is the big magnet, we put the subject in, the magic happening, and then we can get actually images, not only from the brain, but we can image other parts of the body. So a really big advantage of MR, magnetic resonance images, is essentially we can image pretty much any soft tissue inside the body completely safely. So it's not CT magnet, so not CT scanner, it does not have any radiation, and we can actually scan many times during the day, nothing happened to you. So how MRI works? This is probably the worst slide I got. A lot of text. I don't know, bear with me. So, okay, so MRI utilizes physical phenomena called nuclear magnetic resonance. Sounds scary, right? Nuclear. Oh, something wrong with this technique. What this guy is talking about. But this phenomena has nothing to do with ionizing radiation. So it simply says that, the MR can actually measure certain physical quantity of the protons, which is actually part of the nucleus for hydrogen atom. So hydrogen nuclei has a certain physical property called spin angular momentum. So it's essentially a physical name, but this is how nature built the universe. Maybe in different universe there will be something different, but in our universe, hydrogen nuclei has some quantity, which we call angular momentum, and also with this, or simply spin. And this quantity can be actually has associated property called magnetic dipole moment, as long as the spin is placed inside the strong magnetic field. So that's why we need actually magnet for MRI. And because we have so many, water molecules. Each water molecule has a 2 hydrogens atom, H2O, right? So we have plenty of those spins inside us. So what actually happening, if there is no magnetic field, so I'm standing here and talking to you, so I'm not inside the big magnet, although our planet has magnetic field, but very weak. So I'm theoretically inside the magnetic field on planet Earth, but this field is so weak, so all those spins or hydrogen atoms are completely randomly oriented within me, within you, within the water tube. So essentially, I don't have any measurable signal. But however, if I would put myself into strong magnet, suddenly I induce some order inside those, randomly oriented spins. Some of them are aligned parallel, and some of them align anti-parallel to this external magnetic field, which is an MRI machine, which is this strong big magnet. And this causes something macroscopic, which we can measure. And this is actually what we call magnetization vector. Because essentially what this magnetization is essentially we taking all those spins which pointing out, subtract all those spins which pointing down, and the difference is what we can measure inside MRI machine. And in order to measure this, we have to deliver also radio frequency energy to the spins. So we can essentially induce, we call it physically, a resonant phenomena or transition. And that's why it was called nuclear magnetic resonance, because it's actually associated with this angular momentum which sits in the nuclei. But it has nothing to do with, nuclear bomb or anything like that. It's just magnetic properties of this nucleus. So, by the way, this word nuclear was dropped for medical purposes, and that's why MRI is called magnetic resonance and not nuclear magnetic resonance imaging, because people, especially when this phenomenon and early MRI machines were built in 70s, have a very bad associations with the word nuclear. The Cold War, nukes, now North Korea now, so it's essentially, we don't want to hear that, right? So this is exactly the reason why we call it magnetic resonance imaging. But surprise, surprise, for this discovery of nuclear magnetic resonance, there was actually a Nobel Prize awarded in 1952 to both physicists, because this nuclear magnetic resonance has the enormous application not only in medicine, but in biology and chemistry. And it's incredibly important techniques, not only imaging, but actually NMR itself. So, as you probably remember, to get the signal what we can measure in MRI machine, we need the magnet, strong magnet. So I'd like to, this slide tells you how complicated this magnet is and how heavy it is too. So actually, we have here 3 Tesla magnet like this, a little bit newer generation, but still they actually weight of each magnet is like 12 tons. So I mean, it's like 24,000 pounds. So it's pretty heavy magnet. So how we generate such a strong magnetic field like 3 Tesla, which is like 1 million times stronger than Earth magnetic field, we have to put a lot of stuff inside the magnet. And this stuff is very special because it's special material called superconducting material, which can actually carry electric current without any resistance. So what it means, essentially, we have no resistance. Say, for example, if you have old electric bulb in your house, you run electricity through this bulb, you cannot really touch this bulb because it's so hot. And the reason it's so hot, we generate the light in this bulb, electric current is flowing, but also is producing a lot of heat. Inside the magnet, we don't want the heat, so we have to use the different materials. So we cool down those materials to very low temperature, like few kelvins, which is like temperature in the space, with liquid helium, which is very special, liquid gas, really. And then at this low temperature inside the magnet, the current can flow constantly without interruption and produce strong magnetic field. And that's what is used actually to produce measure signal for imaging. And by the way, this picture was actually taken at NIH in 2004 when my first 3 Tesla whole body system was actually decommissioned after 6 years of successful operation. So, but to actually get the image, we need something else. Of course, magnet is important. But say, if I would put myself in the magnet and don't have this something else and measure signal from my brain, the magnetization, I would get, say, signal like this, which would not distinguish, for example, position from water from this part of the brain from water from that part of the brain. So this is actually like a cartoon used in the very famous paper when actually magnetic field gradients was actually introduced, showing that essentially, if you'd like to, this is like a tube of water, and this is another tube of water. So if I have only one field from the magnet, I cannot distinguish this tube from that tube, and I got the signal which is actually has no dependence on the position. However, if I generate additional magnetic field inside the magnet, which varies linearly as a function of space, and we call this gradient, then, we modify basic equation for MR, and then suddenly we can distinguish those 2 tubes, and we got 2 independent signals for the essentially tube on the left, which would be this one, and tube on the right. And this part is called magnetic field, it's called gradient coil, and it's hidden inside the magnet. So if you're going to be scanned in our location, in our place for the research purpose, you are not going to see this, don't worry. But you also don't going to feel it, don't worry, because this is like additional fields are completely harmless for you. It's just in the air. So you don't really see it, you don't really feel it, but we have to generate additional field to get actually image. And surprise, surprise, another Nobel Prize just for this invention. People realized about this magnetic field gradients, and the guy who actually realized that we can use this for imaging got immediately Nobel, I mean not immediately, but he got the Nobel Prize, he deserved, for sure. And this is actually another person from England who actually apply this magnetic field gradients technology for, we call it slice selection, and also invented super fast imaging techniques which we utilize in fMRI, functional magnetic resonance imaging, which I'm going to talk in a few minutes. And there it is. So unfortunately, you know, Paul is deceased. He died a few years ago, but he was actually visited us at 2007, so I took that picture with the Nobel Prize winner. Why not? So this is actually, you saw this already, but this slide also tries to point out that MRI machine is not only the magnet, has, you know, this hidden parts like a gradient coil, but also have a lot of electronics around it. So we actually, in order to do MRI, we need 3 rooms really. One room for the magnet itself, where you go inside this room and go inside the magnet. There is also scanner control room, which I'm going to show you how it looks, where, you know, the operator sits, talks to you, gets your brain pictures, and there's also room there where is a lot of electronics. And we also have a dedicated actually cooling system for the magnet, so to keep you really, you know, calm and safe. So this is how it looks in reality. So this is really MRI machine, all these three things put together, not only the magnet. So if we have all this together, then we can actually produce all those beautiful pictures, which I actually show you some of them as animation on my initial slide. So bottom line is, with the MRI machine, we can actually, in, say, 5 minutes or so, we can collect information from the whole brain, and inside the computer, we can store this information and then use computer to slice the brain in any way we want. So our typical slices are called axial, so we cut the brain in computer, of course, so it's no surgery, nothing like that, it's just in computer. So we can actually produce slice like this. We can also produce, it's a coronal slice, so it looks like this. We cut the brain like this. And then also we have a sagittal slice, which then we cut brain like this. So this is also a picture representation, what is needed actually to get the image. So again, we have to deliver a radio frequency wave, like a, you know, like a radio, because the actual resonant frequency for our scanner is 127 megahertz, which is like FM radio station, a little bit more. So we deliver RF energy. With the gradients, we pre-select a certain slice from the head, and then we collect the data in the presence of this special encoding gradients, which we call, you know, with the gradient coil. And then we collect this and do the magic in the computer and produce the images. So this is actually MRI principle recap. So essentially what MRI does, it measures distribution of proton spins in water across the objects, and various vast different types of contrast can be generated essentially with this technique. And it's completely non-ionizing, it's utilized non-ionizing radiation. So it's very safe. It's just RF waves, which are actually a lot of them here. So you don't even know, you're sitting in the audience and there's a, you know, broadcasting stations everywhere. So if you would have the magic glasses to see RF waves, there will be like, you know, whole room will be full of it. So that's exactly what we use in fMRI. A little bit, you know, stronger waves, but essentially this is what it is. And we have a special device where we detect the signal. We call this RF antenna or coil. And then, you know, we use the magnetic field gradients to distinguish different parts of the brain in terms of the water content and properties. And then essentially we apply something what we call pulse sequences to get the image. And again, MRI is very safe. And it's essentially, we can scan people many, many times, as long as they don't have any metallic objects inside the body. But there's no other really dangers from the MR. So what we can use this, as you can figure it out probably, we can peer inside the brain. So I'm showing you extremely high quality and resolution brain images, which we're getting here. And this is actually, right now, to be frank, MRI is probably one of the most important medical devices used in the, right now in the US and the whole country. Pretty much every hospital has an MRI machine. And in most cases, they have 1.5 Tesla systems, which is, our scanners are twice as strong. So, and what they use them for in the hospital mostly is for structural imaging, to see if there's any abnormal changes inside the brain, like a tumor, like some abnormality, but they don't have a capability for running this at such high resolution. But anyway, so this is actually, again, structural imaging, so we can get the whole brain information. I'm showing you only one slice, but really we have many slices like this in 10 minutes or so. And then essentially there's tons of applications what we can do, but essentially I just showing you this as the eye candy, you know, how high quality it is, how detailed information about the brain structure. So clearly, you know, this is the gray matter again, this is the white matter, this is actually dark as CSF. And there's, I'm not going to show you and talk about the detailed structure and small parts of the brain, how they actually name and call, but we can, this is potential application what we can actually use the structural image. But the remarkable thing about the MR is, you know, but like a flipping and, you know, changing few things on the computer, I can generate completely different looking contrast. So this, yes?
Question, when you do the different contrasts, do you have to run the scan again?
Yes. So this is actually, you know, the previous scan takes 10 minutes. In order to generate contrast like this, in most cases, I have to run another 10 minutes. But there's also like a new techniques right now that you can actually run a little bit longer, not 10 minutes, a little bit like 50% more time, like 15 minutes. And then you can collect at the one experiment multiple contrast. And then in computer, you can essentially produce different contrasts. And that's, in the matter of fact, I'm going to show, this is another contrast which actually maps the vessels inside the brain. So we can, for example, map the detailed map of how vasculature looks like, really. But also, this is what you ask. We can also, in computer, if we have you know, two different image contrasts, we can actually combine them, produce the artificial contrast, for example. But also, you know, once we have the data, we can do things like this. So we have to do, for example, like a rendering. So it's essentially, you know, we already collect in 10 minutes whole brain information, and now in computer we can do things like that. You know, it's not really to show you how fancy this is, but it's actually, this is actually very useful technique for the, especially analysis of functional data. So we can produce surfaces, you know, and then we can essentially split them, rotate them. And this is, again, living brain, this is data collected from the living person. So it's not like we cut this brain, remove the brain, and then do this, you know, with the video or something. It just, this is actually collected like a week ago here at LIBR. So and now, so far I was talking about the structural imaging. So, you know, that we can peer and look on the brain structures, but now let's talk about the functions a little bit. So how we can explore brain functions and how brain is organized in functional domain. So luckily for us, there is a technique which allows us to do so. It's called functional magnetic resonance imaging. So what is all about? So this essentially, in a nutshell, we can make a movie from the brain. And then how we do that? So this is structural image, which you just saw, you know, many examples before. And again, to get the whole brain information for this image, we need typically 5 minutes. So again, I'm showing you one slice, but think about this, you know, we have many slices available. I just, for the illustration purposes, I'm just showing you one. But now, you know, the guy from the second Nobel Prize slide from England realized, there is some analogy with some other branch of physics that we can maybe scan much faster and use the much stronger gradients and do much faster special encoding. So instead of spending 5 minutes per volume, maybe we can do this, say, in 2 seconds. So instead of 5 minutes for, say, 70 slices, maybe, you know, we can collect 40 slices in 2 seconds. What we have to give up something, and what we give up for that is essentially spatial resolution. So, you know, on this image, in the smallest part of the image is essentially 1 by 1, I approximated, by 1 millimeter cube. So, you know, very small elements, like a pixel in the camera on your picture. But on this image, you know, the smallest element of this image is actually a little bit larger, so like 2.5 by 2.5 by 2.9 millimeter cube. So, however, the big difference is to get the volume corresponding, whole brain volume for this type of image, I just need only 2 seconds. So what I can do if I have a capability like this, I can simply repeat these measurements. So this is measurements, my first 2 seconds, I am collecting 40 slices like this, and then in another 2 seconds, I collect another 40 slices like this, and so on, so on, so on, so on. So suddenly, you know, what I can get from that? Here, I have 3-dimensional information, an x, y, z, because I have many slices. But here, I also have 3-dimensional information regarding the space location, but also I have information because in time now. So now what I can say, I can select, for example, certain brain regions right there, and then watch what happens with this brain region as a function of time. And we call this time series. So essentially, it's like a making movie. So I can, you know, pre-select certain brain regions and see what happens with this region as a function of time. And then guess what? Lucky for us, you know, if we put the subject in and run this fast sequence, and then we ask subject to do something. So what happens with MR signal? There is no task, essentially, to the subjects during this data collection. The signal is supposed to look like this. You know, it's kind of flat, you know, varies up and down a little bit, but not much. But say, for example, let's, you know, flash the subject with some visual stimulation. So I told you about the visual cortex on the back. You know, you see some color stuff here. And actually what happens, so nothing happens, and then, you know, the stimulus is shown here as a gray bar. So that means that essentially, you know, whatever visual stimulus was, it's active here. After a few seconds, MR signal goes up. And then stimulus went off, MR signal goes down, and so on, so on, so on. So what it means, luckily, very luckily for us, we have something inside us that task actually affect MR signal. So we have a way to essentially map and measure where brain is being activated during the task. And this is actually a historic part for fMRI, because this is first ever really collected human imaging at Medical College of Wisconsin in 1991. So this was the first observation in humans of the fMRI-induced task activity during the essentially finger tapping. So the subject was inside the MRI machine, and as I told you, we make the movie from his brain. We select the part of the brain, slice like this, so it's not, it's a little bit, we call it oblique because it's angle, which covers the motor area, which I was showing you before. And then subject was asked during this experiment to move fingers like this on one hand, and then on another hand, and then toggle, on, off. So left on this image is right there. So, you know, you see suddenly in the motor area cortex that MR signal goes a little bit up, so it's a little bit brighter. So that means the subject is actually tapping those fingers on the left hand, then stops, and then right hand, and so on, so on, so on, so on. So this was really first, because we call this actually technique blood oxygenation level dependent contrast. It sounds very scientific, but actually, you know, it's kind of simple, really. But although, to be frank with you, we don't really understand this contrast yet for 100%. But what really happens when the neurons actually activated, they need oxygen. So how the oxygen is delivered to the brain, we have a vast network of vessels. You know, I show you this network of small capillaries. There's actually inside those vessels, there's actually red blood cells, and they carry oxygen. There's oxygen attached to them. And the guy who actually, you know, understands the structure of hemoglobin got the Nobel Prize too. It's this guy. So what actually happened when the hemoglobin is fully saturated, then magnetic properties of the hemoglobin, causing that hemoglobin is not interacting with magnetic fields much. So those line is just like MRI's magnet, and this is essentially, hemoglobin, the magnetic field is this way, so there's weak interaction, nothing really changed. But when the oxygen is actually detached from the hemoglobin, there is a big interaction, and this hemoglobin actually affects the local magnetic field. But this is not only what's happening, because when, again, the task neurons are activated, there is increase of neuronal activity somewhere, and then essentially there's also local increase in the blood flow, which essentially change oxygenation of the blood in this area. So, and this effectively caused that MR signal goes up because we have more fresh blood, fully saturated blood, which does not interact with magnetic field as much. So we were lucky because of how, our blood is essentially made, that we have internal contrast in our body, and then we can measure, the signal changes induced by this contrast. So we don't need to inject anything, like some artificial, not healthy contrast for our body. It's just inside us, really. So that was the big discovery, and the guy who discovered this in animals, in rats, his name is Seiji Ogawa. This is my prediction for another Nobel Prize very soon. So, okay, so sure, yeah, so we can make a movie and we get some signal changes, so what we can do with that? So actually we can do some, basic data analysis and statistics and essentially try to see where those brain regions actually are activated given a task. So for example, again, this is our movie, so we're making snapshots every 2 seconds from the whole brain. So imagine we have one condition where there is something happening in the brain. So the signal in this area during this condition is a little bit higher than, say, in another condition that there was no task. So essentially what we can plot essentially for this first 2 second signal like this, another 2 second signal like that from this region, and another 2 second signal like that. And then, we disengage the task, and there's a different signal value. So what we can do, we can compute average here, and compare them. Questions?
Just quick, so in terms of generating the image on the bottom, do you do a slow scan to get that high resolution? And
Yes.
then do the task?
Actually, this is actually structural image, which is independently acquired.
Okay, so
Yes. But what I'd like to also point out here, those images I acquired, say, 10 years ago. They are actually ultra-fast imaging every 2 seconds. But this is also image which looks like this one, but it's acquired now, here, in LIBR. You see how big difference is? So it's essentially also super fast acquisition, but much higher quality of the image. So technology actually improves quite a bit, but we still need the essentially high resolution structural image acquired independently. And then if we can actually do some statistical approaches and some mathematical approaches to actually map this difference and essentially assign some number, and to this number we can assign color, like say red colors mean the difference between those, the number characterized the difference is really high, so essentially we can make it red and then put it as on top of the structural image. So that's essentially what we do in fMRI analysis. It's actually way more complicated, but actually to get you idea. And this is how the raw fMRI data looks like. So what you're actually going to see is essentially like a matrix of voxels. So each box is individual brain region. So we have 1, 2, 3, 4, 5 by 5 voxel. So on this axis within the single box we have MR signal, and here time. So you can see the brain was activated. This is again motor area, left and right. So this was actually when we were testing our magnets. So that was again finger tapping, left and right. So you can see signal goes up when the person tap his finger, and then goes down, and up and down every 10 seconds or so. So, and this is actually example, so this is this activation mask overlaid on top of the high structural image. But the point of this image is, I was telling you about the gray matter, that essentially gray matter is packed with the neurons and where essentially thinking is happening. So this actually shows you that although the bold fMRI is not direct measure of neuronal activity, but because of those capillaries are very close to the neurons, so essentially those activation maps sits directly on top of the gray matter. So we are not 100% on where the neurons are, but super close. Okay, so this is, I may skip that because this is actually, bottom line, we're getting a lot of data from our experiment. So per subjects, typically we're getting like 2 gigabytes of data, but we actually about to start the study on the kids, so, and we will be getting actually 130 gigabytes of data per subject. So there's a lot of data. So we have an fMRI, so we actually have to deal with that. So this is also, this slide is essentially pretty complex, but essentially telling that our data, what we're collecting in fMRI, they contain important information how brain works, but also they have a lot of noise. So it's, every measuring technique, what we do in, real life with any experiments, there's always noise in the data. Even if we build equipment, because we use the copper wire and equipment sits in the room with certain temperature, there's always noise because this is the nature of our essentially lives. There's always something which distract our measurements. So we have a lot of noise in the fMRI data. So not only we collect, for example, fMRI signal, but we collect all the other additional information, how the subject breathes, how the heart is beating, to suppress this noise in our data. And brain is never really, again, addressed. So what this shows you activity actually inside the brain when subject is doing nothing. So essentially we put the subject inside the magnet and tell him, sure, there will be no really task, but essentially you just watch on the screen, there's like some kind of cross to fix your eyes and think about nothing in particular, and this is actually what we're getting. So essentially, you see, it looks like a random kind of variation, but in fact, if you notice, this is not really random. So it's essentially, there's so many brain areas which are co-activated during the rest. So this is actually reflected by, say, for example, yellow color. It's very similar here, and those colors reflect the similar activity across different brain regions. So even when there is no direct instruction to the subject during the experiment, there is a lot of ongoing activity in the brain, which is actually coherent in time and also coherent in different areas of the brain. And this is actually a very important part of research because, we call this phenomena resting state networks. We discover many different resting state networks which actually tells us how the brain operates and works. So for example, in psychiatric disorder like depression, schizophrenia, this resting activity is vastly affected. And we have a pretty good understanding now, and we characterize many networks. So here, I'm showing you like 10. One important one is called default mode networks, which people believe is essentially relates to our self and how we process emotion, many other things. So if you essentially, if subject develop psychiatric disorders, those networks are affected, and also interaction across those networks are also affected. So I'm getting closer to the end. Question?
When someone is meditating, have you shown an MRI how the brain actually slows down?
Yeah, there's actually quite strong research about the,
it actually slows down?
Well, it's hard to tell if it slows down, but clearly affects those default mode resting networks. Yes, there's a big effect of the meditation. Yes.
In a good way?
Oh, I think it's a good way. So, yeah. So yeah, the well-being, it's also, especially meditation is actually very good for you, I would say. So there's actually, even they put the Dalai Lama in the MRI machine, so there's actually a really active ongoing research focus on, what actually happened and we try to meditate. Of course, in the scanner environment, it's kind of hard to meditate, but it's possible. Yeah. So, I hope I show you that MRI can be used for actually peer inside the brain and look also a little bit on more dynamic information, how brain works in time. And also I'd like to show you one more thing which we can actually utilize MRI. So it's essentially we can actually use fMRI to try to control our brain and to self-regulate our brain. And we call this technique real-time fMRI neurofeedback. It's just, I will have on the one slide, I'm almost done. So essentially it's a little bit busy maybe again, but what we can do so again, we're making movie, right? We put the subject in, we scan very rapidly, and we're getting actually data during the scan. We process this data in computer very rapidly during the experiment, and then we can, for example, pre-select certain brain regions which we are interesting. So this is actually on red, is called, shows left amygdala, right amygdala, and this is actually brain region which actually produce, it's engaged in mathematical computation. So we can pre-select information from this brain region, in this case left amygdala, then process it very rapidly, and before the next data collection for next volume for brain starts, we can actually convert this signal, MR signal, to some graphical representation. Like this moving bar. So the bar up means large signal from amygdala. Bar down means lower signal from amygdala. And then we can feed the signal back to the subject. So we can form the feedback loop with the subject during your experiment. So essentially what we can do with this, we can train the subject to control his own brain, at least try to control his own brain. And actually some people, why bother, right? So, I mean, it's actually, there's some technique called biofeedback, which was used before MR, that you can essentially use, for example, your heartbeat sound as a source of feedback, feed it back, convert to some, the sound pitch, or you can use other, like a physiological signal, and then try to control it by yourself, essentially. But with the brain, the big advantage is, especially with fMRI, that we know where we are, so we can pre-select certain brain regions which, for example, may not really work well if you develop, say, depression. So, and we know that, for example, amygdala, there is actually, the brain. So if I would say some virtual, like a pen, and I put my pen through my eye, and then put another pen through my ear, amygdala is in intersection. We have two of them, actually. So amygdala is very important in emotional regulation and many other things, too. But the message here is, we know, based on other research, that amygdala actually works a little bit, response from amygdala is a little bit too weak to positive stimuli for people with depression. So the people with depression tends to have, we call it emotional bias towards the negative. So essentially, then amygdala, you tell them something good and they will make it bad. So, and then amygdala is essentially a response from too weak. On the other hand, if you tell them something bad, then amygdala is on fire. It's just too much. It's working too high. So what we can actually try to do with this is essentially, show the patient signal from amygdala, and we try to teach the patient to correct this we call it hypoactivity to positive stimuli. So essentially, the patient is seeing his own signal from amygdala, and we call the patient, try to upregulate the signal from amygdala to this target bar and try to recall some positive memory during this process. And believe or not, we actually show that this actually really works well. So we're actually helping people with depression. So it's actually beneficial for people with depression, reduce their symptoms, and actually make them a little bit better. It's not that we cure them, but at least we're helping them. And this is the way, you can think about the application for this neurofeedback things for many other things. And actually lots of people are running now neurofeedback experiments and try to use this to enhance cognitive ability, to enhance memory, and different psychiatric diseases, trying to essentially use the different brain regions. It's very hot topic research right now. But this is actually what fMRI can be used for. So essentially, we can really put you in your own brain control with this technique. So, okay, almost done. Take-home message, very simple. At least I hope that you will associate MRI and functional MRI now, that this is actually non-invasive and very safe techniques that allows you to peer inside the brain and also to study the brain activity in normal, abnormal states. And living brain is never at rest. It cannot be simply turn it off. You go to bed, you sleep, you have dreams, and brain is constantly active. It's constantly on. And brain activity is affected severely by psychiatric disorders. And also actually be nice and care about your brain too. So this is just like a message to you as well. So I'd like to thank you for your attention, and I'm very happy to take any questions you may have. Yes?
You said be kind to your brain.
Yeah.
What can we do?
Exercise. Yeah, and don't, alcohol is not too good, especially avoid stress, especially if constant stress is really bad, so, it's just, I highly recommend exercising, walking and not, obesity, try to avoid, sugars and stuff, I like Coke, I guess, maybe Coke is not too good, so it helps you, on short term, you feel better, but actually it's not maybe too good for your brain as well.
Does alcohol really kill brain cells?
Oh, there's actually, yeah.
Well,
But good news is, even if we're aging, there's still, in certain brain areas, there's still neurogenesis going. But I would say alcohol is not as, frankly, we have so many brain cells. So, even if some of them die, brain has enormous plasticity. So essentially, we will still operating and we will still function. But clearly, alcohol. If you drink too much alcohol, it's not good for you.
And do those brain cells, when they die, but do you grow new brain cells?
Well, that's actually, certain brain regions, they actually show evidence that they can regrow the neurons, but it's not really clear if all brain regions can do so. So that's why, you better be, modest, I would say. Yeah.
I've been reading a lot about embodied cognition lately and how we hold our bodies and what our posture, our body-mind connection. So I'm curious about if the MRI, you're lying down and, examining the body that way, if there are testing mechanisms for looking at the body in different postures or the effects of long-term seated postures.
Yeah, actually, clearly, on the physiological level, how the heart is beating, how hard, it's very different if you sit and if you lay down, right? So, actually some MR vendors produce MR machines where you actually sit, so like this. But they are not as good as, our magnets where you actually, lay down in the magnet. But the whole body-mind interaction is one of the main interests of the LIBR now. So there's actually, specialized brain areas like, say, insula, and there's actually, we call this interoception. So there's a lot of active research. So clearly the mind is not isolated from our body, right? So it's essentially just. But from the experimental point of view, clearly there's a difference if you sit and if you essentially lay.
Well, even if you're sitting in a C curve versus an open posture, what the vagus nerve and all that. So is that kind of work going on here?
Well, not in MR, right? Because we are limited essentially, right? But on the outside MR machine, we have another modalities like, for example, EEG, so we can actually assess to a certain extent this. and also through behavioral measurement. Inside MR, you know, we actually kind of stuck to the position what we have, right? Yeah. Okay.
With the studies going on in the connectome,
Mm-hmm.
is the architecture of brains such that, is it possible that, let's say my brain, as it connected, might connect in a different architecture than someone else's, and as a result of it, I might have more limitations, possibilities?
Well, I was about to show you the Einstein brain here during this lecture. The Einstein, for example, Einstein brain has a little bit different structure, but you know, this is hard to tell. There's no, I mean, there's not really like a solid. You know, the purpose of the Connectome project was essentially map connection, you know, within the brain using the very advanced technique like this. I did not even mention during my talk. Essentially, we can also map. We have an ability to map how water diffuse inside the brain. So essentially, through this, we can actually really trace those fibers. So one of my early slides, you know, showed these very colorful maps of the fibers and connections. But I mean, so this project went very well, and you know, map. To some extent, but again, if you compare, for example, small fibers obtained with MRI techniques and also with optical, you know, if we have an ex vivo brain, we can cut it, we can stain it, so we can look on very small details. So MRI may be not there yet, you know, so we can map the main fibers very accurately. And of course, you know, every subject is a little bit different, so it's not like, you know, like we can produce the templates, you know, but I mean, individual, you know, capability. You know, if you have a neurological problems, for example, you got the stroke, so then clearly this can actually damage certain physical connections in the brain. But in psychiatric disorders, we don't really see clearly, you know, damage to the structural images. There's some subtle changes, but not like, you know, in stroke, for example, like brute force differences. So it's actually, you know, what I would say, MR is great, but has its own limits too. You know. Yes.
You know, it's more neuroscience. It's not your research here, I'm sure. I work with children and adolescents a lot, and then I have 6 daughters, and they love to travel. So this is more of kind of a timely question about the Zika virus, and I think we're all pretty familiar with how it devastates a fetal brain. But, and I know that the Zika virus is cleared quickly from most adults in a few weeks. Do you have any thoughts of what a new Zika infection would do to a child who's also developing pretty quickly and an adolescent? Do you have any thoughts of where that's going to take us? Is there research?
Oh, so if I mean, essentially, yeah, it's because,
young brain development.
yeah, I mean, clearly there's a lot of plastic changes in the brain during the development, right? So new connections form, you know, there's synaptic density changes, you know, for example, prefrontal cortex develop, you know, until 21. So you've been bitten by the, you know, by the mosquito, you know, if you are 9, 10 years old, that may have some evidence, but I don't think there's actually any research evidence so far yet. No, we don't really know. It's rather, you know, unfortunately, kind of observational question. If someone finds anything, then we can maybe, you know, put this individual on the scanner and see what actually happened. But we don't really know. And you know, I don't even want to speculate. It's really like experimental. Experiment needs to show that. Yes.
What part of the brain would show genetic disorders?
Oh, well, that's also quite complicated, actually. So, I mean, you know, many factors can affect, you know, our gene expression, environment, stress. You live in big town and everything. So, and but I mean, the changes in the brain are not sometimes obvious too. So maybe on functional level, essentially how the brain regions interact with each other, we may see some associations with gene expression. Although there is some, I guess, some structural evidence too, but I guess,
Is there a certain portion of the brain that?
yeah, certain areas, yes, yes. Yeah, but it's not, I mean, it's, actually the associations on the genetic research and imaging research is actually pretty growing now, and it's actually being, we have even here at LIBR we have actually a few research projects which are actually trying to assess this in, for example, in anedonia, and we're trying to see if there are any associations with genetic expression and some changes in the brain structures and also in the brain functions, but it's actually, but for this type of research, you will need a really huge data set, essentially, of the huge data set of subjects, essentially, to draw any reasonable conclusion. We have a big study here called TALSA 1000, so it's 1,000 people are being scanned and assessed on many levels, so that may actually give some hints soon, hopefully. But actually, realistically, for the genetic research, you probably need like 10,000 or more to draw some. And this is actually, but actually, the general trend in the neuroscience research right now is actually like a big data. So people try to merge, experimental studies in one big data set and essentially look and apply some a little bit more advanced data analysis techniques to draw some meaningful prediction and conclusions. So, but genetics is, it's not easy to really assess well. Yes?
I remember reading that functional MRI would pick up when people have actual auditory stimulation like listening to music, and the same areas lit up when they were just thinking about the same music.
Yeah.
Reading your expression, make sure that I'm not talking about something so silly, because that actually may be true.
Yeah, it is actually true.
So I was wondering if that might also be the case for thinking about motor activity. Like if you can't tap your fingers because you've had a stroke, can you just think?
Yeah, we call this brain-computer interface. It's actually being done. So, people actually, there's several groups who are actually exploring this, for say, if someone lost a hand, right? So there's actually, they actually use not even MR, but even some cheaper techniques called EEG, so electroencephalography. So people are thinking about the motion, and essentially the pattern of the signal, which is being measured by EEG, can be converted to drive the device, which actually replaces your hand, for example. This is actually happening now. Yes. And this actually neurofeedback technique, which I showed you in the last slide, is one of the example brain-computer interfaces, we call it actually.
Seems a lot more pleasant than tying up the effect to the
Yeah.
other side.
Yeah, and actually imaginary, especially in motor activity, that's also very common experiments what people actually publish. Common. There is some publishable evidence that you can actually really, by thinking that you're moving your fingers, you can actually activate the motor cortex. Yes.
So getting back to my question about architecture, we are habitual in nature. So let's say there's a correlation between us forming a habit and the architecture that supports that habit. Then you want to get rid of that habit. Let's say, just for the heck of it, biting fingernails. And the architecture's in there. Let's say the circuitry or the wiring. I don't know if I'm using the right terminology there. Or the network. So how do you go in there to change the habit and maybe substitute
Actually,
healthy habit?
in most cases, I think it's possible as long as you don't have a neurological damage. Because say, brain has enormous plasticity. Even, we all age. So not only kids can learn quickly, even at older times, we still have, plasticity available to us. So what it means, if you develop habit, what really happens is essentially, the network activity within the brain, which reflects your habit, was affected somehow. And then, as long as you do not have a damage to the element of this circuit, you can retrain your brain, most likely, and actually get rid of this habit. Because essentially, you don't need to really rewire physically. You just need to retune your circuit a little bit.
So reconnect those.
Yes. So it's actually, there's evidence that there's fast plastic brain changes. What this really means, the plastic synaptic densities, you know, the way how the neurons communicate, can be affected and can be actually very rapidly changed. So even there's actually evidence from the actual exercising, like juggling,
Yes.
you know, then essentially in a few hours after this, there's actually measurable change in synaptic density in the brain. Yeah. So we have enormous, and also this is actually the reason, you know, why we need the brain, because we live in such a complex environment. We constantly adapting, you know, to this environment. So it's not like, you know, you acquire one skill, bam, you're done. No. So it's essentially, if we wouldn't have this plastic capability of the brain and this enormous amount of neurons and enormous possible connections, then our life would be essentially miserable, I guess, and we couldn't survive.
The synapse, the 10,000 synapse concept, some can detach
Yeah.
and reattach.
Yes, exactly. I hope I convince you MRI is cool. Thanks.